Good morning, everyone, and thank you for being here today. My name is Corey. I am the Global Medical Affairs Lead for External Engagement at AstraZeneca, and I welcome you to our ISV virtual mini symposium titled AI and Vaccine Development. And thank you for taking the time to join us this morning or afternoon. Over the next 2 hours, we'll be looking at approaches that are being used across the vaccine development lifecycle, from early discovery and antigen design to analytics and potential prediction, and ultimately the evidence that we are bringing to regulatory decision making. Vaccine development has always balanced the scientific rigor and speed and uncertainty that is currently out there. And AI is not just automation, it gives us new ways to look at that complexity, track viral evolution, optimizing antigens, anticipating clinical outcomes, and increasing the consistency and interpretability of the data. And so a question I've asked myself, and hopefully many of you guys have as well, is what is AI today? That seems to be changing on a daily basis. And more specifically, what does it look like in the current context of what we work on, right? Vaccine development. So today we'll be hearing from some experts on some pieces of that and to understand what that currently looks like for different parts of a clinical program, right? Whether we have a specific product, we're looking for that product, or we're bringing that up to the public health authorities and regulatory bodies. How do we go about that discussion? And at the same time, many people are also thinking about AI and what they think of as generative chatbots, right? And while those are useful in certain places, the potential for a vaccine toolkit is much broader and also purpose-built for the scientific needs that we have currently and the challenges that will continue to evolve. So today we'll hear some concrete examples, like such as antigen and epitope design to inform structural and sequence models for immune escape and manufacturability, analytics that kind of stratify the responses and sharpen, the hypotheses that we currently have, and also the predictive models that kind of support eligibility, dosing, and endpoint selection while preserving the interpretability to our regulators. While we also are looking at all these amazing things, we want to address where AI is currently also not fit for purpose. So there still is data readiness and bias and reproducibility for some of these algorithms, and how we explain it is is something that we need to work on to build trust with scientists, clinicians, and regulators. So often the most reliable path is kind of hybrid, right? We want to combine that data-driven model with knowledge, enforce constraints, and to keep the outputs as available and auditable as possible. So with that, our goal for this session is to really separate that signal from the noise. And with that, I would want to really bring from the stage our first presenter today. And speaking on AI and machine learning, automating structure prediction and viral evolution, it's my pleasure to introduce Kumar Singh. Uh, and Kumar, apologies, I can't really say your
Yeah, no
last name.
worries.
But from Novavax today to kind of walk through some of that, some of the things that they have been doing at their company. So with that, give me 1 second, Kumar. Let me pull up your slides for you and
Thank
let
you.
me know when you could see it.
Yeah, we can see.
Great.
Cool. Thanks, Corey. Thank you, everybody, for joining. And it's a privilege and an honor to be here, this august audience and a reputed organization like the International Society of Vaccines. My name is Kumar. I work for Novavax, and I have a privilege of leading a very small, very talented, very focused team that's dedicated in making protein-based vaccines. It's on their behalf that I speak and talk about some of the technologies that we are using, including the new ones today, and how we're deploying it to make next generation vaccines. Next slide, please. It's just a brief Introduction to who we are. We are a global biotech company focused on developing R&D assets and establishing partnerships to maximize the efforts that we are engaged in creating new medicines for new vaccines for protecting humanity. We have been around for a few decades now as a company. We have the privilege and honor of making two vaccines that have affected positively humanity on two occasions. One was, of course, we did our bit in bringing humanity out of the COVID lockdown with our protein-based vaccines. And then the other one, malarial vaccine, which was co-developed with our partners in UK, it has saved millions of lives, especially vulnerable populations, kids in Africa. Our vision, why our scientists are encouraged and motivated to come to work every day, and our mission is in the bottom. What we want to do essentially is leverage our science, our technology, and our scientists to innovate, collaborate, and tackle some of the world's most severe medical challenges. Next slide, please. And what do those challenges look like? Is shown here as a short snippet of the pandemics that have happened in the last 150 years or so. Some of them have been, as you all know, and I hope agree, that some of them have been existential challenges to humanity, in very, very big global scales. And we often believe in no news being good news. But given that we have no idea what lies on the right-hand side of the slide beyond what is shown here, I think we owe it as scientists to be prepared as best as we can to address some of those unforeseen challenges. We can talk at length at the slide, but I think suffices to say that diseases have been there, diseases will come that we know about, some we unanticipate, we don't know what comes next, but the science and using AI, I think we can in the future be better prepared to address those quickly. Next slide, please. So this is how sort of aspirationally I and our team look at AI when we talk about artificial intelligence. On a daily basis, when our scientists come to work, we often look at it as a combination and as a partnership of scientific intellect where artificial intelligence meets human intellect. And it's a thing where we see AI to be a friend, a colleague, and a partner in expanding the productivity, but also shortening the lifespan of our discovery activities, which by design, by serendipity, by the very nature of the work we do, it is prone to fail many, many times. And the faster we fail, the faster we succeed in ultimately making a vaccine. And this is where I think the real effect of AI comes in really handy, because in the history of vaccinology, the identification of a pathogen and then subsequently deploying a protein-based vaccine traditionally has taken decades. Today, we are observing biological complexities. Yes, but also we are looking and decoding it in real time. And this is something that even a few years ago would have been a hypothetical, theoretical exercise. So the ability to do this in such a transformative way, but also so fast, is not only mind-boggling as a scientist, but it also entails the credibility of the scientists who are developing these platforms, these large language models, these weighted matrices that they're developing in such a fast and yet reasonably accurately, right? So next slide, please. And the way I have seen this effect so far in our company, and we've been doing it now for a little over a year and a half, is we're currently focusing on the recombinant protein-based nanoparticles, which is one of our key strengths of the company, where we can make these protein nanoparticles that are very highly immunogenic and give you very specific immune responses on a specific disease context. And I'll leave it to our colleagues in Sweden someday to talk about the similar innovations happening on Matrix M adjuvant, which, as you all know, is very safe, very tolerable, and also very effective in generating immune responses and taking the antigens to the lymph nodes where much of the action happens. Next slide, please. And that's the one slide that I will briefly present some of our initial thoughts about how we see AI and HI combining to create vaccines at a reasonable pace. We are now getting some benchmarks with and without AI as to how quick we can be in deploying this pipeline, but also how effective we can be in generating not just desired immune responses, but actually exceeding the levels that you would need in a real world context. So typical pipeline, I'm assuming this looks not that different from many of the big pharma, startup biotechs, et cetera, that are deploying something similar. And the AI component, largely in our experience, has been on the top half of the slide, where we typically begin by our leadership or our partners prioritizing a disease or a vaccine requirement. And we first begin, as I'm sure many of you as well, is you deploy the GPTs and the large language models that have come about with a fairly fast maturity. You put all your PDFs of that disease in one drive, you upload them to Anthropic, Google, Microsoft, Chat, OpenAI, whatever, and then you ask them to do a very deep assessment instead of you and your team of scientists doing it a few weeks. In a few hours, you start getting some decent responses. There's still some element of hallucination still there, but it is going down at a pretty rapid pace where you're now starting to rely on it for some accuracy. What we have noticed, what I have noticed at least personally, is that increasingly in the last maybe 2 or 3 months, AI has now started to argue with you on a scientific basis and tells you that, if you choose antigen X, then yeah, you'll get immune responses, but they'll likely not be as neutralizing as possible. So there's a better choice of antigens, there's better. And then of course, you as a protein engineer, a protein designer, you can now start deliberately choosing those antigens, but also arranging them in a certain order to get multivalency, to get breadth of protection. So from that choice of antigens, you move on to the sequence space where you start creating these MSAs, the multiple sequence alignments. There's one that I'm reminiscing today about a big pharma back in the COVID lockdown days when they started doing this in a sort of small but very deliberate 3D way. So you would start with an antigen against a particular pathogen, and then you would look for variants. In this case, I'm showing 3. You could take 5, maybe 10 variants of the disease. And then you would start with looking at the conservation of that particular antigen. And then you could start building the third dimension, which is both natural but also simulated emergence of new variants against a particular strain. So in this case, the 3 strains would evolve in sometimes predictable but oftentimes unpredictable ways. And there's 2 patterns that emerge in that sequence analysis fairly quickly. One would be in the context of COVID, for instance, how is the virus evolving in a naive population? Early days of the pandemic, for instance. And then there's a differential way in which the virus is now fighting against a vaccinated population or a pre-exposed population. And you can start seeing those patterns emerging, and then you can make some educated, calculated guesses as to what sequence should be as part of your protein antigen to give you the most breadth of response in a hypothetical context. And then you move with the choice of sometimes 5, sometimes 50 clones or construct designs. You then move into the 3D space where you would start looking, and again, there are very popular softwares out there that do protein structure predictions, and you would want a battery of these structures that you have, the sequences that you have generated in the previous block or the square. You would want structures to be generated for those in a fairly streamlined, these are typical standard Python coding things. And then you could look at it in a very similar way that the industry has looked at historically, where you would look at the standard ways of stabilizing the protein antigen. If you find a hole, you mutate it, an alanine to a phenylalanine. If you find alternate charges, you put in your acidic residues, glutamate to aspartates. Some of them are pretty drastic. Others are much more subtle. And sometimes we have seen in the past where you can even addition of a single carbon, for instance, valine to isoleucine often has effects of that. Now, that's where, at that point, we start our experimentation process, where you start making those constructs and testing for expression and stability. And in parallel, what is increasingly also becoming standard is now you have a predicted antigen structure. And then can you use the other AI structure prediction tools to predict antibodies? Because what you want to know is your antigen that you're generating, is it folded? Is it predicting the right epitopes? Is it generating the type of surfaces on the top of the protein that will actually instigate the immune system to recognize a danger signal? And that experience in our hands in the AI/ML mAb profiling has been fairly limited, but it is non-zero. We have started to get successes where we actually can make antibodies fairly quickly using the predicted sequences from these AI tools. And after that, of course, the standard way, you do your fairly rapid cloning. In the past, we have done it in 24, 98 wells. If it comes to a pandemic with a 100-day mission response, you could scale it up in theory with fairly low cost to 1384 well plates, for instance. And then you can also in parallel screen for expression, stability, and purification protocols. And then because AI has now predicted things and you've done things in parallel, you quickly come to the bottom of the slide where you're now characterizing the protein antigen and asking a question like, is it any good? Do you have the corrected folded protein? Is it stable at 37 degrees for weeks, sometimes even a month if needed? Because that gives you the confidence that in a real world setting with the cost of goods pressure and everything else, your protein will likely be a good vaccine. And that is where, at least again, in our company, we have a very good privilege of partnering very early, even in discovery stage, with our AD, PD, and formulation and process development colleagues. Because when we think of things in milliliter scale, they think in 50-liter scales. And there are enough opportunities there to start optimizing the processes very early on. And that gives you obviously an advantage in both time and also understanding what does that regulatory environment look like. And typically what we have noticed with all of these pipeline engagements and optimization, that once this pipeline is fully operational, you would take anywhere from 12 to 18 months. And now we're having some direct comparisons as to what does AI actually do in expediting that journey to the clinic. Where AI, in my mind anyway, is still very much in its infancy. There are some efforts happening is on the in vivo, in vitro evaluation. I think that's where the data sets are not large enough yet, where you could get accurate predictions of these individual weights that define these large language models. And that is understandable because you would need very large data sets. And I think that is where there's a food for thought here for all the pharma partners, those listening in, those that share in our passion of creating new vaccines, is probably the best way. One way of going forward would be to form a consortium where you can have AI look at the individual data sets of these companies, preclinical as well as clinical, and come up with more sophisticated, more accurate models that can be shared with everybody. And then, of course, we typically are now at a stage as a company where we are looking for partnerships in driving the future clinical development of these programs. So that's a very quick read. I'll stop here and hand it back to you, and hopefully we'll have a good Q&A. Thanks, Corey.
Thanks, Kumar. Really appreciate your time today to kind of review that, like very succinct, but really good kind of transition now into our next presenter, who will be Thomas Trolle. Building on discovery and the structure that we're talking about here, Thomas will be presenting on how AI platforms are being used to identify novel vaccine targets, including polio, and how generative protein design approaches can improve antigen optimization. So kind of going into the process that Kumar was talking about and the earlier portion of that landscape, I'm going to hand it over to Thomas. And Thomas, I believe you're going to be presenting your slides.
Yes, thank you for that introduction, Corey. Can you see my slides?
Yes, I can.
Perfect. Yes, so yeah, thank you for that introduction and for everyone for showing up to this really nice webinar on how we can use AI and machine learning in vaccine development. So I'm from Evaxion, so we're a small company located just north of Copenhagen. Yeah, we use AI and machine learning at the core of what we do with regards to vaccine development. So at Evaxion, our core technology is our what we call our AI immunology platform, which is essentially a collection of various computational and AI-based software tools and workflows that we can use to understand, decode the immune system and decode various infectious diseases with the overall goal of designing an efficacious and protective vaccines. So at Evaxion, our AI immunology platform, while it is our core technology, we of course have a lot of disciplines outside of bioinformatics that goes into helping us develop our platform as well as our pipeline, ranging from everything from disease biology, preclinical development, CMC, and translational medicine as well once we get into the clinic. So we have a pipeline that's centered both around in the oncology space as well as infectious diseases. And for the purposes of this talk, I'll be focusing on the infectious disease arm of the company. But as you can appreciate from the slide, we do have a number of preclinical candidates in this space, quite a few in bacteria. So we have a Staph aureus candidate, gonorrhea, as well as an undisclosed pathogen that has been licensed by MSD. And in the viral space, we are also developing a vaccine candidate against CMV. So as I hope you guys will all learn from the webinar today, you can use AI to solve a lot of challenges within the vaccine development space. And at Evaxion, we've chosen two use cases that we focus on specifically with our platform. So the first is target discovery. So I'll also be showing you how we can use AI to identify and rank novel targets to include in our vaccines. And then second of all, once we have identified some promising targets, we also use AI to design or modify or optimize the antigens to sort of ensure that they work better once they are in the vaccines. So to give you guys an idea of how we do this with our model, so I think like Kumar's slide, and the first step is of course always deciding upon which pathogen it is that you want to pursue developing a vaccine for. And then for our platform, what we actually use as input to the platform are then simply the protein sequences of the proteins that are expressed or found in this pathogen of interest. We then have two arms within our platform for infectious diseases. And the first is our Eden arm of the platform. And this is our arm for identifying B-cell antigens. So the first thing that happens within the platform is that we take these sequences, run a suite of different AI-based prediction tools that then profile each of the antigens or each of the proteins found in the pathogen and sort of featureize it. So we can begin to understand. So the model can begin to understand, what different features do these proteins have. And in this case, an example could be sort of which epitopes, both B cell or T cell epitopes, are predicted to be found within these antigens, but it could also be other things like more topological features, like does the protein maybe have some transmembrane regions, a signal peptide, features like that that can tell the model, where in the bacteria, for example, a given protein may be found, as that can inform the downstream models on whether a given protein could be an interesting target for a vaccine. So as I said, these features are then fed into a downstream AI model that takes in all the features and then attempts to predict how relevant a given protein may be for a vaccine. And this essentially ranks all the proteins that we have in the pathogen, giving us a prioritized list of potential vaccine antigens that we can then start looking at in the preclinic. As I mentioned earlier, not all of these antigens are necessarily ready to include in a vaccine straight out of the box. So for those where it's necessary, we also run some AI-based antigen design in order to finally end up with our final vaccine design. So just to give you guys some data showing what we've been able to do with antigen ranking with our platform. So we've tested the Eden arm of our platform in a number of different pathogens. So here I'm showing data from 4 different bacterial pathogens that have been tested in preclinical infection models. And as I hope you can appreciate from the plots, on the x-axis we have the prediction score that we get out of our platform, and on the y-axis sort of a protection score, you could say. And we see, at least in these different models, we see a fairly nice correlation between how our platform ranks the different antigens in these 4 bacteria, and how protective they actually end up being when we test them in these preclinical models. So based on some of this work, that has sort of laid the groundwork for some of our earliest vaccine candidates within the bacterial space. So the first is our Staph aureus vaccine candidate. This was as I said, the first candidate that we actively worked on in Evaxion. We have a large number of data, but the highlights are that we've been able to show that our vaccine candidate is protective in various different animal models. Included here, shown here, is some data from a mouse sepsis model, but we've also shown similar results in different mini pig models as well. And our second candidate is a gonorrhea vaccine candidate, also a multi-component vaccine designed using antigens identified by our platform. This we've also shown in, once again, a mouse model, but in this case, a vaginal colonization model, that mice receiving our vaccine are once again significantly protected against the infection compared to mice receiving the adjuvant alone. And in addition to this, for the gonorrhea vaccine candidate, we've also been able to show in various in vitro assays how we have quite a broad protection against 50 clinically relevant gonorrhea strains. So then to go back to the model and then to tell you a bit about the other arm, which we call our Raven arm of this of the platform, and this is our focused around identifying T-cell epitopes that we can include in our vaccines. So here, once again, we start with the protein sequences, but instead of calculating all these protective features, what we actually then use our AI model to do is to identify peptides that are presented by MHC or potential, as they are potential T-cell epitopes within these sequences. Using these predictions, we can then identify larger fragments of the proteins where we have a high density of predicted T-cell epitopes within the proteins. And once again, we can rank these T-cell epitope hotspots, and then we can either use these as a vaccine, a T-cell based vaccine on their own, or we can combine them with the antigens that we have identified with the Eden arm of the platform to create sort of a combined B and T-cell based antigen design. And sort of the reasoning for looking into these T cells is, of course, to get the cellular component of the adaptive immune system in play, and also to, you could say, broaden the number of different antigens within a given pathogen that we can include in a single vaccine, and not being limited to, just having a handful of antigens represented in the vaccine. Using this hotspot approach allows us to now include maybe 10 or 20 different antigens in a final vaccine design. And just to give you guys some data showing how this works, we, as many others, have, of course, tested our platform on the SARS-CoV-2 virus. So in this plot shows how the predictions that come out of our platform, shown down here on the bottom, align fairly well with experimentally validated epitope regions of the SARS-CoV-2 genome. So here the height of the peaks is sort of you could say, a hotspot density score of potential T-cell epitopes. And I think, as you can appreciate, the predicted hotspots within the SARS-CoV-2 genome line up fairly well with those that were then end up being experimentally validated. And we took this work further and used our Raven model to design a purely T-cell based vaccine against SARS-CoV-2. And again, in a mouse model, we showed that out of the 17 T-cell epitope hotspots that we identified, we were able to show that 15 of them were immunogenic. And then in collaboration with a group over at UPenn, who had a transgenic mouse model, we were also able to show that vaccinating with our T-cell epitope hotspot-based vaccine was able to protect mice against a lethal infection, challenge with a lethal infection of SARS-CoV-2 in these transgenic mice. Yes, and then finally, with the last 5 minutes, I'll also like to take you guys through some of the newest developments that we have in the platform, and that is around how we are using AI to optimize our antigens for inclusion in vaccines. So once again, you can do a lot of very interesting things with these novel generative protein design tools. But within the context of vaccine antigens, we've once again chosen to focus on two main areas. So the first is for those antigens that, when you express the wild type sequence, are not necessarily very easy to express and manufacture. How can we use AI to optimize this so they can be scaled up and included in vaccines? And the second use case are for antigens that may take on different conformations. How can we use AI to stabilize these antigens in the conformations that are actually relevant for the disease? So I'll start with the first. This is just a case that we have from our CMV candidate, where we had a target antigen from CMV that we were interested in including in our vaccine. However, we were having trouble expressing this protein recombinantly. And we hypothesized that this was due to the fact that the protein in question naturally is part of a larger complex of proteins within the virus. So the hypothesis was that when you're just expressing this protein on its own, it probably doesn't fold well without all the other partners that it's supposed to have in its natural setting. So our idea here was to identify, using structural biology tools, the residues within our target antigen that were involved in protein-protein interactions with the other proteins in this protein complex. And then what we did was we tasked what's called an inverse folding algorithm to generate a new sequence for these amino acids. And for those unfamiliar, an inverse folding algorithm is essentially an algorithm where you give the model a protein structure and task it to generate a sequence that will then fold into that structure. And the way, these tools are trained on a lot of soluble globular nice proteins from the PDB. So they tend to suggest or generate sequences that also fold well and are easily expressible. And that's exactly what we saw in the lab. So when we tried to express the wild type antigen in this case, we got really poor expression. But when we then optimized it, generating a soluble sequence for the protein, we were then able to improve that to a level where we could actually start working with it in our vaccine candidates. And then just to give you a short example of how we can stabilize proteins in a disease-relevant conformation, I think many of you are familiar with these the pre-fusion conformations of various viral proteins. So in this case, in our CMV program, once again, we were interested in creating a stabilized pre-fusion glycoprotein B design, as it is known that this conformation of the GB protein is relevant in a vaccine. So to do this, we once again used various computational tools, structural biology tools, to identify a couple of mutations that we could make within the GB protein. So we essentially added 2 cysteine residues inside the GB protein that would form a disulfide bridge, locking the GB protein in the pre-fusion conformation. We were then able to generate these pictures of the 2 versions of the GB protein. And as you can appreciate here, when we express the unmodified wild-type GB protein, we get this rod-like shapes consistent with the known structure of the post-fusion GB, while adding our cysteine modifications to the protein, we then start to get these more globular shapes consistent with what is known about the pre-fusion GB structure. So this is all well and good, but we also needed to show that this actually made a difference in a vaccine setting. So to that end, we immunized mice with both versions of the GB protein, and we were able to show that looking at the antibody titers, we were able to get good titers against both conformations, actually, or both versions of the GB protein. But when we then actually tested the ability of the mouse serum to neutralize the virus and prevent viral entry into the host cells, we were then able to see that serum generated by the pre-fusion GB vaccine was much more potent than serum from the post-fusion GB. Yes, and that's all I had for you guys. Once again, thank you, ISV and Corey, for the opportunity to present here at the webinar.
Thank you, Thomas. And we have a lot of questions for you later in the Q&A. And some of your Q&A during these sessions,
Yes.
and we'll be answering them at
Look.
And please make sure as you're typing the questions to say who the question is for, so we could get to it later. But thank you, Thomas. And this kind of goes into what Kumar was saying, right? We saw the full landscape. We see how you are creating all these proteins and making it as close as possible to what is needed for a potential vaccine. And this kind of dovetails into our next presentation. And so we will be having Federico from AstraZeneca kind of talking about AI analytics, machine learning for event rate prediction. As we move into the development and execution side, right, we have a vaccine candidate potential in place. Now we're bringing it into the clinic to patients. How is AI a potential tool to kind of help support event rate prediction, understand how many patients we would need in a potential clinical trial when it comes to vaccine development, and how this informs the trial design, risk assessments, and decision making. So with that, Federico, I will leave it to you. And I do not see your slides yet, but let's see if you can pull them up first. Perfect.
Yeah, can you see the slides? Okay. Hi, everyone. Can you hear me? Yeah. Okay. Hi, everyone. I'm Federico Francone. I work as Associate Director of Data Science at AstraZeneca. In particular,
one second. Can you put them into slideshow mode?
Sure. Thanks. Yeah. Okay. As I was saying, I'm Federico Francone. I work within the clinical data science team at AstraZeneca, in particular within the vaccine and immune therapies. And yeah, thanks for having me here. Excited to have the opportunity to present at this ISV Symposium on the use of AI in vaccine development. And today we will be seeing a use case of how forecasting the incidence rate of diseases can actually support study design for clinical trials, and it enables key decision making. So we mostly walk through a prediction like of US RSV, just as a use case and a way to walk through our forecasting framework. Just to give a more detailed picture of what the topics we are going to discuss, we'll first outline the key problem we are aiming to solve and explain why forecasting the disease incidence rate enables decision-making both on study design and monitoring activities. Then again, like this presentation is not about providing insights on US RSV, the models, and the most important predictors to the model, but we use that as a way to walk through our key concepts of the forecasting methodology that I've developed. We start from comparing the final output that we had from this 1 year ahead forecast. So by having the forecasting performance, basically comparing our predictions versus against the actuals of the US RSV season that we could observe afterwards. Then we'll dive deep into some of the key concepts of our forecasting framework, starting to understand how we get a bunch of raw signals and we shift them in time to improve their predictive power. So then we let machine learning models extrapolate patterns from all these time-shifted variables. And we don't only use one predictive model, but we combine multiple statistical and machine learning models to improve forecast accuracy and robustness. And finally, even though this looks like a complex, big ensemble of different models, we'll show how we can actually interpret and explain it. So let's start with point one, the key problem we are aiming to solve. So in vaccine efficacy studies, the key problem is knowing far in advance if and when we will have enough events for the study to be appropriately powered. So we like knowing this far in advance. So if we were able to forecast the study events, we could enable decision making for the study team, such as anticipating the readout date in case there is an optimistic prediction of the study events. Or on the other hand, we might enable decisions on adjusting recruitment plans. For example, enrolling more subjects by opening more sites to increase the probability of reaching the desired endpoints. So we have developed a sequential framework. The first part consists of forecasting the real-world incidence rate. So as we can see in the top chart, and that triggers a simulation framework to estimate the study events. So this forecast, incidence rate forecast, directly translates into the study events estimation. Let's dive deep a bit more into the problem. We first have to highlight that in other therapy areas, it's reasonable to assume a constant attack rate for the control group. But however, in infection diseases are adding an additional layer of complexity compared to their therapy areas because of this seasonal incidence rate, which means the attack rate is not constant over time, but it changes every season in terms of peak magnitude, peak timing, so start of the season, end of the season, and so on. This complicates things, and that's why we need to forecast it far in advance in the future to plan how many subjects we should enroll in the studies and for the study to be sufficiently powered. If that's not clear, maybe from a pure theoretical point of view, let's see that through an example by referencing the right chart. So let's assume we are at in the mid of December, and we have enrolled this many subjects so far. And we have also observed from the middle chart, we can see that we have observed these many events up till mid-December. Now, the key question we are aiming to answer is whether or not we are going to hit the target events by readout date. So in this toy example, the readout date has been set to July. And whether we are going to hit or not the target events depends on the incidence rate. So assuming that so far we have observed these incidents, that's why with the forecast we can answer this question. For example, by referencing this example, we can see that by having this shape of the US RSV season, we would hit the target events even earlier, right? So this will enable decision making in terms of anticipating the readout date. Otherwise, again, we can open in more sites and increase the probability of reaching the endpoints. So that's been clarified. We have developed a forecasting framework, and we have used initially for a big phase 3 trial involving low attack rate and therefore high sample size. That's for a respiratory disease in the US, so US RSV. But the key point here is that we were able to forecast it accurately more than 6 months in advance. And the same framework has been currently used to predict other disease incidence rates in other countries. So actually it's reusable, the same methodology. Let's dive deep a bit into the US RSV. From before diving deep into the modeling aspects, let's highlight the main characteristics of this incidence rate. As we can see from the left chart, on the y-axis, we have the hospitalization rate per 100K. And we can see that over time, by already observing that the seasons are very heterogeneous, here they differ much in terms of peak magnitude and timing. We can see especially that post-COVID, the seasons are becoming much larger and much more inconsistent than pre-COVID ones. In terms of seasonality and timing, I think that's more clear from the right chart, where we have the overlaid seasons compared against each others. The blue curves here represent the pre-COVID seasons, and we can see that they share similar patterns, similar shape, and the peak magnitude. And as mentioned earlier, post-COVID, the seasons start to be much more larger and inconsistent. The purple curve represents the latest season from last year, which was the purpose of our forecast. So it was our target to predict. And we can already observe that the shape is quite unusual, right? So, for example, this right tail of the season never occurred in the past. Generally, the shape is so different that if we try to forecast it by assuming it would be the same as any of the previous seasons, we wouldn't have any luck. So that being clarified, let's see how accurate was our forecast, which we predicted at the beginning of the season. So before the season starts, this purple sign is around mid-September 2024, and is where actually we refresh the forecast, which is represented by the dashed line. We can compare against the actual season that we observed afterwards, and we can see that we are mostly capturing the old shape in terms of peak time and peak magnitude, and also the right tail, the unusual right tail. The only error was by forecasting an earlier season by a couple of weeks, and later on we will also be able to explain why. What I can anticipate now is that to have accurate predictions, that's more kind of a general concept. Especially for forecasting, we need the good predictors, so good variables in input. And in our case, these are mostly represented by other incidence rates, especially from Southern Hemisphere. So we don't provide just a static forecast. Actually, to enable key decision making, we are constantly updating our forecast every week to account for the changes through the seasons, both in the target variable itself, so for the US RSV incidence, but also to react to the changes in the predictors. We can see that in this animation. So the vertical line, if we stop at any point, represents when the forecast is produced. So it means that we can refresh the forecast by using only data prior to this date. And yeah, again, the dashed line is our projections. We can see how the forecast accuracy highly changes over time. And particularly, it starts to be much more accurate by end of August or beginning of September. Let me stop it in two random points. One, before August, we can see that forecast accuracy in terms of peak magnitude was not very high when we were around July or August. But when we start to be around September, it starts to be much more accurate. And that's not due by chance. That's actually due to the fact that we use a lot of incidence rates, including from Southern Hemisphere and South America. And South America seasons are finished, are complete by end of August or beginning of September. That's why our forecast is able to incorporate these changes and to reflect that into the final projection. Now, the key question is like, how do we select these predictors, right? The ones that I've been mentioning. How do we select the top variables to use so that the machine learning models can actually forecast into the future? So let's start diving deep into the forecasting methodology and explain how we search for these predictors. The very first step is once we define the target variable, in our case was again this for this respiratory disease in US RSV for the target age group that we were planning to run the phase 3 trial on. We start from conducting a literature search to identify what from literature seems to be a factor that is related to our target incidence, right? For example, weather, as many of us know, many respiratory diseases tend to peak in winter. So that's for sure a factor that we might potentially incorporate into the model. Also, other respiratory incidence rates in other countries and demographic groups are all factors that, from a theoretical perspective, could actually be included in a potential model, right? But the problem is more complex than that, because, of course, for anything that could work theoretically, we also to reflect that in practice, which means we need to verify and validate that this data is actually available and also weekly refreshed. Like it has to be updated over time and publicly available. So once completed this step, we end up with a bunch of potential predictors to the model, but not everything goes into the model. We have a sequential feature selection step in which we first identify the leading indicators which are those signals that are consistently anticipating what actually occurs later in the target variable. Because in forecasting is really relevant to leverage some signal that is constantly anticipating what happens after, right? If you can see, if you can reference the right chart, we can see the blue curve being our target variable, so the US RSV, and the orange curve being the RSV in Chile. And we can see as the RSV in Chile is constantly anticipating what happens in the US RSV, both in terms of seasonality and also trends. As we can see post-COVID, where during COVID, there was, of course, as we all know, it was an anomalous period, so there were zero cases. Then the trends start to increase, and that's actually reflected and anticipated by the RSV incidence rate in Chile. So that's kind of an example of a leading predictor. It's worth to highlight that we are not looking for causal relationships. So it's not a necessary condition that the input signals have a direct causal impact with what we are trying to predict, as long as there is a relation. So for forecasting, for pure forecasting goals, objectives, it's enough to have, let's say, correlated predictors, even though the actual root cause that is relating the two signals might be attributed to some latent factors, but that's out of scope for the pure forecasting goal. Another example of leading signal can be seen from Georgia, infants, so different age group, different US regions. We can see that the orange curve is a bit more noisy than compared to Chile, but is still anticipating by a couple of months what happens in the US, while Chile was actually, by being in the southern hemisphere, was anticipating by 5 or 6 months. So that's just to give the main idea of what is a leading predictor, a leading indicator. Once we define a few steps, we end up with a set of signals, then we shift them in time. We'll explain this concept later on in the slides. And we let machine learning techniques to select, let's say, the optimal set of time-shifted variables. So basically, for the case of USRSV, we started from more than 5,000 potential raw signals, and we ended up with a bit more than 30 high quality, so high predictive time-shifted variables, which are then fed into the model to extrapolate the patterns from the interactions and forecast into the future. A few more highlights on the selection of these predictors, since as mentioned earlier, it's a key point to forecast, to have accurate forecast. We can mention that the very first step is actually evaluating the data quality for each of these raw signals. So in terms of data availability, granularity, and we want to ensure that we don't have to deal with noisy signals. As we can see, for example, in RSV in Belize, from the top chart, we can see that mostly there is no relationship already from a visual perspective, and probably the signal seems to hint that in the last couple of seasons, the data was not even refreshed, probably, right? But anyways, there is no strong relationship between this raw signal and our target variable. Then again, like we are searching for leading predictors, we are shifting them in time, we are creating multiple time shift for each leading indicators. And for that, I've developed a custom framework based on mutual information to identify the optimal shift and whether a signal is leading or following our target. Just to give an example of a following indicator is US flu, right? The orange signal, even though it seems to have to be related with our target, is actually not very useful for forecasting because maybe it's not fully visible from these charts, but is consistently happening a couple of weeks later than what we are trying to predict. And even conceptually, is reasonable to assume that something that happens after what we want to forecast is not very valuable for our predictive model. So after defining this set of time-shifted signals, we remove the redundant ones, so the highly correlated ones, and we ultimately let machine learning and statistical methods to select the optimal set of predictors. We use both linear and nonlinear methods, but here probably is out of scope for this presentation to dive deep into the specific methods that we are using. But I'm happy to answer any questions related to them. Let's go a bit more in detail into the time shifting part. We can see again by referencing US RSV in Chile, how time shifting the predictor increase the relationship between the two. And this relationship is, in the case of Chile RSV, is maximized when we shift it by 26 weeks in time. We can see indeed that if we stop the animation, from a visual perspective, the two charts, the two curves almost overlap when such a shift is applied. And from a simple metric, that's not what we use in production, but is one of the metrics that we can use to quantify what we are seeing visually on the top chart. On the bottom chart, we can reflect through the absolute correlation how this relationship increases and is maximized when we lag Chile by 26 weeks. What does it mean having the optimal lag to 26 weeks? It means that actually Chile RSV is anticipating US RSV by that amount of time, or in other words, that to forecast US RSV incidence today, we can look back at what happened in Chile RSV 26 weeks ago. That's the key point of having a leading predictor, because it enables forecasting into the future. And it's important that we consider not only one optimal shift for each candidate signal, but multiple time shift. This is a bit more in detail, but just to give the high-level idea, if we shift the signal in time, the two seasons are not fully overlapping. If you can see in 2024, the orange curve is a bit happening earlier, right? Even in this anomalous period during COVID in 2022. That's why you create a multiple shift for each predictor to account for these inconsistent relationships over time between the input predictor and the target variable. So just to wrap up the key points, we are doing that for a bunch of predictors, not only for Chile. So we end up with a set of time-shifted predictors that are highly related with our target variable. And we can, at this point, we end up with a data set that enables fitting machine learning and statistical models. Now, the problem is more complex than fitting just a linear regression by having as a Y target the US RSV incidence in this case, and by having as X variables the time-shifted incidence rate predictors, right? Why the problem is more difficult than that is because in predictive modeling in general, and therefore also in forecasting, we are interested in a model that can generalize well on unseen data, so it can basically be accurate into the future. We are not really interested in a model that can fully capture the patterns, the historic patterns, therefore, in technical terms, a model that is overfitted, but we want a model that is it can fully generalize into the future. And to do that, we apply, first of all, we always hold back some data to validate. So we first train the model in a bunch of data, and we always hold back some data to then validate, choose the optimal model, and finally to test the model on some data that has never seen by the models during the training phase. And also we combine multiple models. Why do we combine multiple models? Is because each of those capture different patterns. And as let's say, each pros and cons. Is a known technique that model stacking, so stacking multiple models, increase and opportunely choosing the best combination of them, highly increase forecast robustness and accuracy. So to give an idea of this part, in this forecasting framework so far, we involve the fit of more than 20 models, ranging from traditional time series ones like autoregressive, like ARIMA, seasonal naive, Holt Winters, and Prophet. And these are the traditional local models. Then we expanded into the machine learning models that can accept a stack of multiple time series in input. So that means that we can augment the amount of data points. So having additional data points for machine learning models is actually beneficial for their training. And that's actually doable and feasible thanks to these machine learning frameworks. Also, we use some deep learning frameworks here without getting very much into detail, are more complex architectures that have been designed indeed to capture and retain memory over time of sequences. And then also I think it's really interesting to highlight that we are using also recently released time series foundation models. These are pre-trained models on billions of data points and seems to be the latest trend in time series. Some of those use the same architectures used for large language models, so for the generative ones. but with the same architectures, basically combining with a set of predictors that we can give in, they're able to actually have a very accurate forecast by combining basically what we are providing them with the billions of time series they've been trained on. So we first optimize individually each model, and after this hyperparameter tuning, we basically find the best combination of models through a cross-validation process, and we end up with, let's say, our final forecast can be seen as a weighted average of different predictive models. Even though now this can be seen as a very big ensemble and very complex, we are actually able to interpret it by attributing the importance to each predictor without getting into detail, because it's a bit out of scope for this presentation, of how we assess and attribute the importance to each individual predictor. We can see that from the left chart, we have the percentage of importance, so the overall contribution of each predictor to the overall forecast. For example, RSV in Chile, on the overall ensemble, on the overall stack of models that we're using, accounts for almost 35% of the importance. Now it's out of scope again to list all the other predictors, what they are, because that's specific for US RSV prediction, and it will change for if you wanted to predict other diseases in other countries. But it's worth to highlight that besides the absolute importance, we are also able to break down the contribution and the effect of each predictor on the overall prediction. By having defined the overall prediction, the one on the top, we can break down indeed the effect of each predictor, for example, of this X2, a generic specific predictor into the model. The green areas are positive contributions to the forecast, while the red areas are negative contributions, which are lowering the forecast. And we can see that, for example, our right tail of US RSV incidence, we were able to capture that mostly thanks to Chile, as we can see, because indeed from March to May, that's the green area, so the positive contribution Chile is having on the model. So now let's wrap up a bit the part on the forecasting side, the methodology. Hope that we clarified a few aspects of how we develop this framework and how we leverage Southern Hemisphere signals to predict the Northern Hemisphere. Let's go back to the original question of predicting study events. So as mentioned in the very first slides, we have this incidence rate forecast, and that translates directly into an event estimation by triggering a Monte Carlo simulation framework to have these study event estimates. Now, how this probabilistic framework, simulation framework works is out of scope for this presentation, which indeed was supposed to be more about forecasting. But again, the key point is that by having this accurate long-term forecast, we enable key decision-making on study design and monitoring activities. So to conclude, again, it's critical to have to know far in advance how many study events we are going to expect in the study, because indeed that can drive key operational decisions, such as anticipating readout dates or increasing the number of subjects enrolled to have to increase the probability of reaching the desired endpoints and for the study to be appropriately powered. And in our case of US RSV, this framework delivered a very accurate forecast, as we can see on the bottom chart, despite the seasons being very heterogeneous, both in terms of peak timing, peak magnitude, and overall shape of the season. And especially having a structural break after COVID that really made it this whole problem much more challenging than pre-COVID. So we are aiming to apply the same methodology to forecast other disease incidence rate in other countries. That's also what we have been expanding to, that's already been part of the current progress. And we are also aiming to expand to increase the set of raw signals to give an input to these models, not only including other disease incidence rate for other respiratory diseases, but also including other type of signals such as vaccination rates and disease strains. That was all from my side. Thank you, and I look forward to receive questions.
Thank you, Federico. wonderful presentation, and kind of dovetails really well into what we've been talking about so far, right? So understanding how to build the vaccine, now making sure that we are considering the pathogen at hand to understand when to run a vaccine clinical program in humans. This goes into our next topic, and so we will be having Guilhem from EpiVax to talk about EpiVax's FDA experience, which includes perspectives on NAMs and MMFs and how AI-enabled insights can be translated into regulatory confident frameworks. One thing that I want to point out, and sorry if I mispronounce your name, but Javier had asked a question, and I think Guilhem, you're going to be talking about this, but there is a scientific movement in trying to eliminate the animal model to test vaccines and biomedicines by instead using computer and or AI models. What is the current status of this? So as you're going through your presentation, I think you could definitely touch on this piece with the new methodologies that regulators are looking into. So with that, I'm going to pass it off to Guilhem to give our last presentation of today.
Great, thank you very much, Corey. Perfect pronunciation of my name. So yes, I'm Guilhem Richard, the Chief Technology Officer at EpiVax. So thank you to the ISV organizing committee for inviting me speak at this mini symposiums. So yes, I'm gonna talk about a slightly different topic, still on AI ML. But once we generated all of these results with the new models, how can we include them in regulatory submissions? And oops. There we go. So here's the agenda for my talk. So I'll have a little bit of an introduction, giving some background on AI. We've seen a couple of applications already, but just so we're on the same page, and how we're using AI at EpiVax for vaccine development. And then I'll switch gear and talk a little bit about some of the regulations that are coming out. More specifically from the FDA, I'll be touching base on a couple of documents that deal with using AI in regulatory submissions and what the regulators are expecting. And lastly, I'll touch base on our experience following one of those pathways, which is the model master file. So a little bit about EpiVax. If you're not familiar, we've been around for 25 plus years. Next month is going to be our 28th anniversary. And we've been doing AI ML, starting to do AI ML at a time when people thought it was really crazy to use computers to predict anything about the immune system. Even when I started working at EpiVax 10 years ago, I was getting some pushback from time to time by people, by me presenting data generated using computer models. And now when you go to a conference, you can't have a presentation without mentioning AI ML. And so what we're doing at EpiVax is to develop and provide various solutions focused on immunogenicity assessment. So that includes both in vitro and in silico approaches for assessing immunogenicity, and with applications in both the biotherapeutic area, helping designers of monoclonal antibodies or multispecific antibodies, as well as supporting vaccine designers for optimizing the designs of their antigens. So just to start fresh again, what is AI ML? You've heard some examples. I feel this is a topic where if you're asking 100 different people, you're going to get 100 different definitions. So at least to me, artificial intelligence is really the broadest term that encompasses everything that you're doing in terms of modeling. So AI is really any computer system that performs quote unquote an intelligent task, and that task can still be in a way a dummy task. It can be a rule-based approach. If a variable have a specific value, then do this. And a great example is using Excel, for example, to format reports, highlight specific areas of a table based on different values. So it doesn't necessarily need to be very complex. Then you go into the realm of machine learning. which is a subset of AI, where here you're starting to develop models by training them on real life data. So now you're moving away from your fixed rules. And if you're thinking about a real world life example, that could be your recommendation system in your preferred streaming services. For example, you watch a lot of specific types of movie. Well, based on what you've seen, you may like these other movies. Then there's some type of distinction that goes between ML and deep learning. So sometimes we refer to deep learning as a subset of ML that uses complex models. And the difference in complexity is typically done when you start using neural networks, again, to analyze what is complex data. It's not necessarily terms that are very well defined. But here, typical examples are going to be image processing. So, for example, facial recognition and language processing. For example, your Google Translate app that will translate English into any other language. And then you have what I think a lot of people think about when they hear the term AI, which is generative AI, which is here again a subset of deep learning that focuses on generating new content. And here you have all of your favorite chatbots as examples. So applications of AI in the vaccine development. We've heard three talks providing a great diversity of areas where AI is being applied. So it's not only something that can be used or applied in early discovery for identifying new antigens or predicting epitopes. You can really apply these approaches throughout the development pipeline. So again, early discovery, selecting your antigen, performing immunogenicity or safety modelings, understanding how you need to design your trials, for example, identifying the best dose or trying to understand how you're going to stratify your patients. And that can also be done in the manufacturing CMC steps to either enhance the production of your vaccine or also to monitor a specific aspect of the productions. And some key considerations is really the first one, the most important, I think, is that the quality of the data, of the training data, is key. There are many different types of models. You have what we're calling, quote unquote, more simpler model or more complex models, but it really starts and ends with the quality of the data that you have on hand. And using fancy or complex models won't fix issues that you may have by using a training data set that is not necessarily well curated or well adapted to the question that you're trying to address. The other consideration is whether also you're working with a model that uses continuous or static learning. So in continuous, the model continuously learns from your data that you're generating. So when you run the same model at different time points, you're not necessarily getting the same results. And which is very different from the static learning, where once you train your data, you essentially lock it, and every time you run it, you get the same results. And one is not necessarily better than the other. Both have advantages and disadvantages. And again, it's really dependent on the specific question of interest as to which type of algorithms or models makes the most sense. And lastly, I think it's important to note that, at least for now, AI is not necessarily fully replacing your traditional vaccine development methods. But these AI approaches are really valuable when you combine them with existing methods for optimizing your antigens or your trial design. So the way we're using AI at EpiVax is, again, to focus on immunogenicity assessment. Here we typically use two core algorithms. We use our EpiMatrix tool to identify T-cell epitopes that may bind to either HLA class 1 or HLA class 2 molecules. And depending on the type of analysis we're working on, we can go from a global analysis assessing an entire antigens to focusing on specific regions of that antigens that contain what we're calling epitope clusters. And the other major tool that we're using is our Janus matrix tool, that once you identify putative HLA ligands, how are they going to be seen by the immune system? Are they going to be seen as foreign and induce an immune response, or are they going to be seen as self and be potentially passively tolerated by the immune system? And then by combining these two concepts together, that can help us provide a refined understanding of the immunogenic potential of specific sequences that can be quite helpful if you want to rank specific antigens of interest for developing vaccines. And we see, like many other groups, we see these core tools as quite adaptable. And then we can use them in various workflow that will address specific questions. So we have designed various web-based platform that we use when working on different fields. Either if we want to assess the immunogenicity of biologics, we're going to use one of those platforms, which we call ISPRI. Generic peptides are going to be a different platform, and so on. And also, we reformatted these tools when we work in vaccines. And here for this audience, we have two platforms of interest. First one is our IVAX platform. This is our vaccine design platform for what I would call your standard infectious disease vaccines, where you can triage antigens, identify immunogenicity hotspot, concatenate different epitopes together. And here I've just showed a couple of publications that include results from our IVAX platform. And we also have another platform that we call Answer, and this is more geared for personalized immunotherapy. So identification of neoantigens and personalized cancer vaccine design, and again, with a handful of publications tied to this platform. So now that you've used AI to develop your vaccine, whether it is early stage, late stage, or for designing your clinical trial, how do you include these results in regulatory package? And really, how does the FDA at least view the usage of this information into when they're reviewing your regulatory packages? So I'm going to touch base briefly on 3 different documents that were put out by the FDA in the past years, each focusing on a slightly different aspect of AI. But as you will see later on, there's still a very central theme that is common across them, which relates to explainability of the model, the usefulness of the model, and validation of the model. But the first one is about the use of AI in supporting regulatory decision making. The second one is the new approach methodologies, or NAMs, which is meant to replace, at least an attempt to replace, the use of animal models. And the last one is the concept of model master files, for which we don't yet have a guidance from the FDA, but there has been some publication and meetings on this topic organized by the FDA. So the first document that I'm showing you here is the FDA's guidance on the use of AI in regulatory decision making. So the FDA think about AI as any computer model. So it's not just your chatbot. It includes also machine learning based models that generate predictions or recommendations that you then use in regulatory documentation. So it is really tied to models that inform a regulatory decision making. It is not applicable to other use cases of AI in general day-to-day business activities, for example. So that's a clear differentiation. And again, why did the FDA issue this guidance? Well, as you can see, there's really an increased reliance on using AI in drug and vaccine development. And it It's very powerful. We've seen multiple examples of this in the past presentation. You can reduce the timeline. You can reduce the use of animal studies. You can really start analyzing complex data that is very difficult to understand when you're looking at it. And in the end, all of this helps us make better decision, develop better drugs and better trials for patients. But at the same time, the FDA is a little bit worried about what's coming out of these AI model. Are these output credibles? Is it transparent? Is it fit for purpose? So my takeaway on this guidance, and I should say that all of this is my interpretation of this guidance. I don't want to speak for the FDA or other regulatory agencies. But in my view, the FDA uses a risk-based credibility framework. Essentially, you need to think about several questions when you're using results from an AI model, an AI ML model. First, what is the question of interest? What's the decision you're trying to make using that AI model? The context of use. Exactly how are you using the results from that model to reach the decision? And what is the model risk? This is probably the most important part of that draft guidance. So here, this relates to two different concepts. How much does a regulatory decision rely on AI? And what happens if the results is wrong? So for example, here you can see you can think that there can be very different scenarios, whether you're trying to optimize the dose for your vaccine, or are you trying to understand who is eligible for being enrolled in a specific trial? And you can see that if you're wrong with one of these results, then that can have very different effect. Are you going to overdose a patient, or are you simply not going to enroll a patient? And at the same time, are you just using results from an AI model to reach to that decision, or is the AI model part of a collection of many other data points that are collected through other methodologies, such as in vitro assays or other in vivo studies? And then, based on that risk profile that you determined, you have to build up some credibility evidence package for that model. So here, this is you have to mention the data that you're using to build the model, to test the model, and to document that the results that you're getting make sense. And the riskier the model, quote unquote, the riskier the model, the more documentation, the more credible the model needs to be. And all of this is still very fuzzy, I think. There's not a clear-cut definition of what is a low-risk model and what is a high-risk model, and how much data you need based on that risk level. Everything is still left to us to define the risk and to define how much data we want to provide in order to make the regulators comfortable with using results from these models. And so here, my key takeaway for the vaccine developers is, again, that higher impact uses need stronger evidence, and that, at least for now, AI is typically not used completely alone. It's typically used to complement other data sets that are part of the regulatory package. The next document that I'm touching base on is this recent draft guidance on new approach methodologies. So this is what we're calling it the NAMs. So these are human-centric methods. So it's not just the NAM guidance doesn't specifically focuses on in silico models. It can be any methods, whether it's in silico or in vitro, that can help us generate non-clinical safety and efficacy data or mechanistic data. But that is really centered on human interpretation. So this is really the guidance and the effort that is put forward to replace the use of animal models. we know very well, and we have known for a very long time, that animal models are not necessarily very predictive of what happens in a human. We have a different immune system than animal models. We have different MHCs, for example, different types of response. We know that some molecules that are safe in animals are going to be toxic in humans. And now that we're having a lot more data that is being generated and that is being publicly available, and with the use of these new models, we can now start to build models, or to build assays, in vitro assays, that are a little bit more appropriate for answering the questions that we want. That is whether the products that we're developing, is it safe and toxic, and or toxic specifically in human and not necessarily in animal models. So this is still a very early draft guidance that can be changed, that may be subject to change. But at this point, it sounds like the usage that these NAMs don't need to be fully validated to be included in regulatory submission, as long as they're fit for purpose and you have some type of validation that rationalize their usage for your application. And here again, the emphasis is on transparency. How did you build the model? Why does it make sense to use the model? And does it address a specific question that other approaches are not appropriate, for example? So here again, my takeaway on this draft guidance is that there are 4 practical questions that you need to answer. The first one is the context of use. What decision does your NAM help make? So again, a lot of these questions are very similar to the questions that are brought up by that first guidance. What is the human biological relevance? Is that NAM, does it provide meaningful data that you can't obtain through another way? The other point is technical reliability. Are you using something, an assay or a model that is reproducible and transparent? Can it be explained? And lastly, fit for purpose. Does it replace, fill a gap, or complement an existing data? So using NAM is not mandatory at this point. Their usage are certainly welcomed by the regulatory agencies, but you still need to have a good reason for employing them. It's not just to remove the animal studies that you didn't want to do. There still needs to be a strong rationale for using NAMs. And as I was mentioned, at least at this one, they don't need to be fully validated from a regulatory standpoint to be used. And again, they're typically not used in isolation, but they're there to complement a data package, a strong data package. And so the last concept that I will touch base is this concept of model master file, or MMF. So you have probably heard about drug master files, or DMF. So conceptually, this is very similar, but it's applied for a model. So these are regulatory documents that can be applied to any models or modeling platforms that have gone, and here I'm quoting the text, sufficient verification and validation. Unfortunately, what that means precisely is not defined. So we still don't know exactly what the FDA, precisely what the FDA expects. But at least it still ties in with the other documents that were put out by the FDA. You still need to have some type of transparency about the models, and you need to have a good validation package showing that the results of these models are valid. And one benefit of these MMFs, so similarly to the DMFs, is that they can be referenced across multiple regulatory submissions. You don't need to resubmit the same validation data package for every submission. You can simply reference the MMF of interest in the application. And so here again, why MMFs were proposed. the usage of AI is again increasing quite a lot. We're using a lot of models, but the FDA was not necessarily knowledgeable of how these models were built, especially if those were proprietary models. And they didn't necessarily know either if these models were sufficiently validated for them to make sense to be included in a regulatory data package. And the effort is also to improve the consistency and efficiency in the FDA reviews. The idea is that those MMFs won't necessarily need to be fully reviewed once they've been reviewed once, and that once you've submitted one, it's going to make the process a lot easier for reviewing the regulatory packages. And so here, again, very similar takeaways as before. It makes sense to use MMFs when you have models that are well-defined, transparent, and reproducible. It's quite helpful to have a platform where you can just submit once your models and everything that's tied to how you built it and how you validate it into one single package, as opposed to resubmitting the same information over and over again. And more importantly, what's very beneficial, at least for us from a CRO perspective, is that it establishes a direct line of communication between the FDA and the group that developed the models. So if the FDA, if the reviewers have any specific questions about information that we include in an MMF, they can directly reach out to the model developer, bypassing the sponsors who are submitting the regulatory validation. And the key takeaways here is that these MMFs are still very early stage. They're not necessarily formally defined by the FDA. There's no draft guidance that has been put forward to date. And right now, they are using the some existing pathways, submission pathway. They're using the Type 5 DMF submission pathway. There's no dedicated separate submission pathways that is specific for MMFs. And as I was mentioning, what needs to be in the MMFs and what the FDA really wants to see in those MMF is still not very clearly defined, and there's still a lot of discussion that goes between the FDA and model developers to see what makes sense and not make sense to include. And so to wrap up this presentation, I'm going to briefly talk about our experience going through the pathway of creating a model master file and submitting the model master file in the context of abbreviated new drug applications. So this ties with one of our platform that we call the Panda platform, that is geared towards assessing the immunogenicity of generic peptide drugs and their impurities. So if you're not familiar with these pathways, drug developers or generic drug developers submit an abbreviated new drug application, or ANDA, to have their generic drug approved. And through this pathway, the sponsor don't need to go through a clinical trials, as long as they can demonstrate that their generic has some type of bioequivalence with the reference listed drugs. And to do that, they must use orthogonal assays to demonstrate bioequivalence, and that can include both in silico approaches and in vitro approaches, a combination of both in silico and in vitro approach. And so we use results from our Panda platform as one of these orthogonal assays to inform on the risk potential of some novel impurities that can be present in the generic peptide drug formulations that may not be present in the reference listed drugs. And so we have set up a regulatory package that includes our models and methodologies on how we tackle this problem and how we define risk categories based on results from our models. And here I've put the overall outline of the MMF that we submitted. There's a background section where we're defining the question of interest. What is the problem? What needs to be addressed? The second section discusses a little bit more about the methods. So this is where we talk about all of the equations that we use to run in our analysis, how we build the models, what type of training data we've used, what type of validation data that we use, and also some of the reports we're generating using those models. Then how are we applying those models specifically for assessing the immunogenicity risk of peptide drugs and their impurities? So here, how are we reusing the results from those equations to provide a risk category for these impurities? We also added a section on software and hardware environment, where how and where we're running all of these predictions. So that helps us support our technical reliability. And lastly, we have a validation section showcasing that yes, the results that we're providing make sense, and all of these individual models were validated, and using them provides meaningful information that can be gained without running full-scale animal models, for example. And as I was mentioning, these MMFs are right now submitted as drug master files, so they need to conform with the ECTD format. And so all of this information is currently included in module 5 of the DMF package. And we've submitted it. It's been a little bit over a year ago now. We submitted on February of last year. As far as I can tell, we were the first company that submitted an MMF to the FDA, again, specifically for focusing on the immunogenicity risk assessment of generic drug peptides and impurities. And we were happy to see that the submission of these MMF was quite in line with the NAM roadmap at that time that the FDA had put forward. And since then, we've actually seen draft guidance coming out for using these NAMs. And so to wrap up, I think we're all aware that AI/ML is really becoming a much bigger factor when developing vaccines and biotherapeutics, not just at the early stage. It really can be beneficial throughout the development landscape, from early stage to late stage development, including manufacturing. And because of this, the regulatory expectations are moving very quickly. You would be talking to me a couple of years back, I would have very different answers for you, at least, or we would have a different view of what the FDA wants to see. So the landscape is really evolving very rapidly, but the FDA is still very willing to see results from those AI/MLs to be included in regulatory applications, again, as long as they're scientifically justified, that they are explainable, and that they are validated in a way that they feel comfortable that using results from these models make sense. And again, as I was mentioning, we've had a little bit of experience with formatting these models for regulatory applications by submitting our Panda approach as an MMF. And here, again, the key takeaway for my presentation is that I don't think we necessarily need to be fearful of AI. At least the more I speak with people from the FDA, they welcome the idea of using AI for streamlining application, for streamlining the review of regulatory application, but using these models in those documents must be accompanied with a clear documentation and validation data package that really makes them comfortable comfortable seeing and understanding really what these models are doing. And I'd like to thank you very much for your attendance. And I think trying to stop sharing. There we go. And I think we're going to jump to the next step of the webinar.
Yeah, thank you, Guilhem. I really appreciate your insights on this. It's really cool to see where we have gotten with AI, but given that we're working with regulatory bodies, a lot of time and discussion with additional companies, and as the programs move forward, they'll feel more comfortable with these new modalities, right? So, since I have you on screen, is there any thoughts, and anyone please chime in from the panel around what we think about animal models going forward? Do we expect that to kind of have a play into things, given the current administration? And we're strictly talking about the US FDA at this point in time, right? So, do you guys think that there is a space for AI to kind of continue to push forward in a sense there?
So I'll start.
Yeah, I'll go after.
Perfect. So I think there's definitely a lot more that we can do with AI. I think it's also very dependent on the data that we have available. And here, this is where getting more animal model data can be helpful to at least see if the models that we're doing can replace results we're getting from these experiments, and if they're also relevant of what we're seeing in the clinic. So I don't think animal experiments are necessarily going to completely go away right now, but certainly, at least from my perspective, it makes more sense to use models or even in vitro assay at this point that provide results that make a lot more sense. I know we've been working with animal models for a very, very long time. Even when I started working in this field, I was showing these results from in silico models, and people were telling me, well, they're not predictive. We need to do animal models, but results from animal models are not necessarily predictive as well, based on the failure of all the clinical trials. So to me, there are definitely going to be more and more influences from AI, ML, but it is going to be a gradual increase.
Great. And Thomas?
Yeah, I agree completely with what Guillehem's saying. And you also had a slide showing your platform for personalized cancer vaccines. And that's also a field that we are in. And already there, I mean, I think there have been several trials where we've tested these, where Evaxion and many different companies have tested personalized cancer vaccines where you're essentially designing a vaccine on the computer without going necessarily doing any animal testing or even in vitro testing. So I think that's maybe sort of paving the way for the use of AI more in vaccine design or drug design in general. And I mean, potentially, and depending on how well those drugs end up working, could also be expanded to more general vaccines and other drugs that are not necessarily personalized.
And to that point, Thomas, since you were just talking, there was a few questions for you when it comes to your presentation, right? So from Bimal, do you consider the considerable variability of the antigen of the pathogen, like influenza, HIV, to design a candidate as a futuristic vision?
Yeah, that's a really great question and something I missed in my talk. But yes, that is of course a very important feature that we look at when we're designing our antigens, and we look on this in two levels. So the first is, you could say, more on the sequence level, a bit like what Kumar shared in his slide. If the same protein is found in different strains or isolates or variants, how conserved is that protein? And also, I think more relevant for the bacterial side, if we're looking at different isolates, how often is a given protein found in those different isolates? Because, especially if you're looking at something like a toxin that may be a very relevant vaccine antigen, those are not necessarily found in all clinically relevant strains of a bacteria, right? So we look at it in that in those two different through those two different lenses, you could say.
Yeah. And when it comes to, and yes, in a sense, depending on the pathogen and what is the most virulence versus the most uh from like whichever way it's presenting, right? So how do you select antigen components for combination or formulation? Is that something you use your the AI models for? This was a
Yeah. Yes. 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 That's a great question from Junheng. And I would say on the B cell antigen side, we're still using, you could say, more a traditional vaccine development approach where you have a bunch of single antigens that you test individually and find those that are effective and try to combine them in a way where you can show that the combination adds a significant additional effect over the individuals. We are exploring different ways of doing this, maybe computationally in the future, but for now, we're still doing it sort of in the traditional way. On the T cell side, we are actually looking at, because T cells are HLA restricted, different T cell epitopes are more or less relevant in different individuals and different populations. So in our the T cell arm of our platform, that is something that we do take into account in order to ensure that we're getting a broad coverage of the target population that we're aiming for with our vaccine. Yeah, so that we do that in a bit of a different way.
Okay. And Thomas, the T cells, that's the Raven model.
Yes, exactly.
So how does it take into account? I think you kind of touched on this, but how does this take into account the high variation in coronavirus?
Yeah. Yeah. So yeah, essentially, what we're doing there is that for each individual T cell epitope that we find in the coronavirus, we then look or in the virus in general, we then look at once again how frequently is that given epitope found across different variants of the pathogen that we're interested in, and that once again can give us an idea of how conserved that individual epitope is, and allows us to score different epitopes differently based on how frequently or how broadly they are found in various variants. And another thing that I didn't mention in my talk, but sort of another benefit of targeting T cells in a vaccine is that you are often able to include epitopes from proteins that are inherently more conserved across variants. So if we take the coronavirus as an example, I mean, I think what we saw was that a lot of the variants was found in the spike protein, right? So if you had sort of a B cell antigen vaccine targeting spike, that was there was a lot of immune pressure on the virus to escape the immune systems of the different people that were vaccinated. So you saw a lot of variants in those, whereas with a T cell vaccine, you could include epitopes from the other antigens or the other proteins within the coronavirus, where you could say the conservation was much higher, thus allowing you to get sort of a broader coverage while across the different variants, but with the same vaccine without needing to update your vaccine.
Thanks, Thomas. And for Guilhem and Thomas, there were a few questions. So if an academic center would like to reach out to you guys to partner, is that possible? And do they send out an email to you guys and just reach out and see what potentially can happen?
Yes, I can go first. So we're very happy to collaborate with academia and other industry partners. So yeah, we can definitely either shoot me an email or go to our website. We have a partnering page where you can contact us, and we're happy to collaborate in different settings. That is a core thing we do at Evaxion, collaborate around using our AI immunology model to develop vaccines with different partners.
Sounds
Very
good.
similar
And
for me. Best way is to reach out to our business department. And if you email me, I'm happy to reach out to connect people with our BD team. And there's also a contact page on our website.
Same here with Novavax too. We're very committed, yeah.
thank you. And Federico, there's a few questions for you in regards to your presentation. So, does the prediction apply to all US RV? So, in regards to your example of RSV infections, right? How predictive is future newborn US RSV infections? I believe you looked at plus 65, right? Over 65 years of age, but have we potentially looked into future newborn?
Yeah, great question. Yeah, as you mentioned, like we targeted this age group because it was aligned with the phase 3 trial that we were designing, but we can apply the exact same framework to predict hospitalization rates for different age groups and demographic group. In that case, maybe like if I share the screen, that helps the discussion. Like from the CDC RSVNet, if you pull the same time series just for different age groups, you can see that the patterns are slightly different. And this for me makes sense because there's been like vaccine program like in the market, right, for smaller age groups in the last couple of years. Reason why probably like the incidence rate has been decreasing more aggressively for infants than for adults. But aside of speculation, I'm not a subject matter expert. In this case, if you had to predict, for example, children between 0 or 17 or different age groups, we will just, in that case, the model and the old feature selection framework will will choose a different combination of raw predictive signals. But yeah, so it's generalizable. We can use the same framework, and it's been already done actually for different diseases and different age groups.
And Federico from Manon, she had a question about why do you think RSV occurrence in Chile was such a good predictor of the US season? Do you look at other countries in the Southern Hemisphere as well, or
Yeah.
and what did it look like?
Yeah, I said at the beginning, I mean, I can share all the details of the model here, but yeah, as we mentioned, like we look at a lot of incidence rates, not only RSV, and especially South America, we saw that is a very good predictor for North America. So it's not only Chile, it's also other countries and other disease rates. And the reason why they might be so correlated and Chile might be anticipating, yeah, it's the key question that, as I said, is out of scope for a pure predictive task. What I, just for curiosity, out of curiosity, I searched back then when I was developing this model is that in the last years also there has been similar campaigns, for example, vaccination campaigns in Chile with concurrent timings, which might affect the transmission dynamics, right? Like in the case of US RSV, the age group was 65 plus, but in case there is a vaccine out there in the market in the last couple of years, as it is in the example of the US, vaccinating infants can change the transmission dynamics and shifting the curve to the right, which now is not visible in the latest season here, right, the 2025. But if you see Chile RSV in the latest season is much lower and shifted to the right, similarly to how the current season has been for RSV. A reason why maybe it might be similar, right? Like similar vaccination campaign in similar timings we can assume that might lead to similar effects. But I think these slides just wrap up the pure aspect, right? Like two signals might be correlated. The root cause, so the latent factors might be a mix of vaccination rates, weather, lockdown measures. But yeah, that's out of scope for the predictive analysis.
Right, right. And also going into it, I guess, a question from Nena, right? So you kind of touched on this, but exploring real time. How real time can we get within season learning? And where models update based on early intra-season RSV activity to forecast the current season's trajectory. How does that compare in performance and reliability?
Yeah, that's a great question. I mean, if I understand correctly, we are wondering whether we might not look only at past seasons, for example, for Southern America, but also for looking at what's happening within the season, right? Like when the season starts, let's say. Is my understanding correct of this question?
I believe that's correct, but
Yeah.
and I think you did show that, right? That like during the season,
Yeah.
it does um you do
Yeah.
update based off the season as it goes.
Yeah.
And then the next question is in regards to past seasonal, does your AI model predict failures when it comes to
Yeah.
past seasonal flu vaccines that had low efficacy? So I guess we're talking about the flu season now.
Yeah.
Do you look into other viruses and pathogens?
Yeah. So maybe like this is shorter, so maybe I can take first the one of flu. For flu, I observed that the model, the forecasting is more challenging because like RSV, I'm not a subject matter expert, but it's known that RSV is much more stable than flu, so it mutates much less. Reason why, if the flu vaccine is not accounting for a prevalent disease strain, that highly affects the incidence rates, and that originally was not included in the model. I've been actually working to include also this as a additional predictor, right? We can always expand the set of predictors, for example, to say, has this H1N3 dominating variant in the US been accounted into the vaccine? If not, then we might then create ad hoc predictors to then give the model this information and also accounting for these strains, which I see that really impacts on both the peak magnitude and timing of the season. But that's an extra layer of challenge I've been facing, to be honest. And regarding the intraday part, that's a very great question. Just want to clarify that when we are forecasting here for all the rest of the year, it means that we basically forecast for almost the next 52 weeks, right? The 1 year is roughly 52 weeks ahead. So the reason why we look at past seasons is because by September, the Southern Hemisphere seasons have been completed. And if you looked at the dynamics that happen within the US, the issue that can be a bit frustrating from a forecasting perspective is that we might struggle then to forecast it. I mean, that can be great for explanatory purpose or for short-term forecast. For example, just to give an example, I mean, even weather was not selected in then as a predictor to the model, that I can say. Because for weather, we have the forecast of temperature and humidity, so all the weather signals for the next 2 weeks. And while that's great to predict the short-term forecast, so for the next 2 or 3 weeks of US RSV, weather is not a great signal to predict 1 year ahead, right? So when we go back to looking at what happens within the US ongoing season, and then we face with this constraint of saying, how do we forecast the next year? And to be just a bit more detailed, the matrix that we're building, I was hiding this slide for time constraints and also because it's probably too detailed, but we end up with an input matrix that has all these shifted signals, right? Like if you want now like to reference 20 of May, Chile, like by 26 weeks, it means that we look at Chile back in 26 back, what happened in RSV in Chile 26 weeks ago, basically. We do that for other signals, for other incidence rates, but then to forecast into the future, we still need the same input matrix. So like if you want to forecast into the future, we need, like the machine learning models take in input this input matrix, and after being trained, they are able to output these expected Y target values. But again, so to summarize, if we look at concurrent signals not leading, then we wouldn't have this the way to populate this matrix unless if we predict these input predictors. But then rather than predicting the target variable, we should do a lot of forecast of all the forecasts of each predictor, so with a lot of uncertainty related. So that's complicated. It's a great idea, but I think it works either for shorter term forecasts or for more explanatory purposes, if I'm making sense.
Yeah, I mean the weather analogy is really important, right, Federico? These are very, very interesting approaches that we're taking. So thank you so much, everyone, for joining us today. I know there were a few questions that weren't answered, and I know that we're over time now by 5 minutes. So I really appreciate everyone joining us today. When it comes to the questions that haven't been answered yet, I encourage you to please reach out to the person that you want to pose that question to, and they will be able to answer that at the appropriate time. So again, I would like to thank everyone for joining us today on our virtual ISV Symposia in April, and would like to hope everyone, including the presenters, for your time today, to have a nice day, and we'll be seeing you in the future with our next presentation. So thank you so much, everyone.
Likewise.
Thank you.
Thank you, Corey.
Bye-bye.
Bye bye.